What system are we in? Respiratory system, still there. Last time we spent a lot of time talking about anatomy, but before we did, we talked about the fact that respiration happens due to 4 processes, really, right? What were those 4 processes? What? So we had a fancy term for that inhale and exhale. What was that? Pulmonary ventilation. It's even on my screen right now, right, guys? So that was one of the things that we talked about. What else did we say was going to be a function that helped us with respiration? There was external respiration and internal as well. Good. And we had transport. So we already just mentioned that pulmonary ventilation is that gross movement of air, right? The inhale and the exhale. What do internal and external respiration have in common?
They exchange.
What do you mean by exchange? What's going to happen?
They exchange gases between the lungs.
All right, we see gas being exchanged between the blood and something else, right? So notice we're also going to be talking about external and internal respiration today then, right? Because I say we're talking about gas exchange. What was the difference? Where does external respiration happen? Between the lungs and the blood. Good. And so internal would obviously have to be what? Between the blood and the body's tissue. So really, we're looking at exchange that happens in those 2 different blood circuits that we've talked about. The external respiration is happening out there in the pulmonary circuit, and then internal is happening in the systemic circuit. But I think it's a good idea to start at the very beginning and focus on that pulmonary ventilation. So let's see how we're going to be moving air into and out of the lungs. We know that we could call that inspiration and expiration, right? Inspiration's just the same thing as inhale. Expiration's the same thing as exhale. And you guys know a thing or two about what's going to help regulate movement of air because it's the same thing that helped the blood to move. How did the blood always move?
From high to low pressure.
From high pressure to low pressure. So it's going to be the same deal here. But the setup here is a little bit different. I mean, think about it. When we were focusing on the blood, we had basically things set up in a loop, right? Because the blood left the heart through arteries, and then we saw the blood entering capillaries where exchange happened, and then the veins helped to carry the blood back to the heart, right? So we saw this consistent effect of the pressure dropping as we moved through that circle. But what's the setup like here in the respiratory system? Let's think it through because we talked about those parts last time. So where's the air going to enter the body first? All right, through the nasal cavity, right? And what else? Or the oral cavity, right? So we have the nasal and oral cavities. And both of those cavities connect to the same tube. What's the name for that full tube? Start with a P.
Pharynx.
The pharynx. Good. Thank you. And the pharynx is going to connect to another tube where the air is going to go. The larynx, exactly. And we know that connects to another tube. A trachea, exactly. And well, before the trachea gets to the lungs, it branches, right? What did that form? right, generally speaking, those are bronchi, but technically they are primary bronchi because they're the first branches. And we know that once they get into the lungs, they're going to branch further, right? So what are the next branches? Secondary bronchi, and then they branch to form the tertiary bronchi, and then they branch to form the smallest tubes, which were what? The bronchioles. And then, well, the bronchioles are going to eventually form tubes that dead end at spots that we call what? The alveolar sacs, exactly. So that's clearly not a loop, right? It has a dead end point. What that means is that we're going to see pressures changing then with this setup, because otherwise, how are we going to allow the air to move into the body and out of the body through these same tubes? So we're going to do that by changing the volumes of things, like the volume or the size of the thoracic cavity and the lungs. And Boyle's law helps us to understand the relationship between the size or volume of something versus the pressure that we find within it. So first off, before we look at the equation or anything, let's remember what we know. What always causes pressure? The molecules that are interacting with something, right? So that's what they're showing us with that picture with the little boxes. The little black dots are our molecules. And that box is the container that they're found in, just like the lungs would be a container for the air. And what Boyle's law tells us is that if we're focusing on a gas, like the air that we're bringing into the lungs, as long as we keep that temperature pretty consistent, we should see a relationship between pressure and volume, where the pressure times the volume that we start out with should equal the pressure times the volume that we end up with. So, for example, if we notice what's happening in my picture, we're decreasing the size of the box, right? So that's decreasing its volume. Then what's going to happen to pressure? The pressure is going to have to increase because there's an inverse relationship there. Remember, if we're decreasing the size of the box, well, now the molecules have less room to move around without hitting something. So at any given point in time, we'll see more molecules interacting with something. And the more of them that interact, the higher the pressure gets. And the opposite is true. If we were to go back the other way and increase the volume, what's that going to do to the pressure? That's going to decrease the pressure that we find inside the box. Are we all clear on that example? Any questions before we move on? It's about to get trickier. Yep. I gave you a different figure for it, but the wording is the same. Well, you can write down the formula. It's pretty easy. So yeah, I might have done a couple of edits on the slides, but most of it is on yours. All right, so what's going to help us with changing the volume or the size of our container? Muscles are. And different muscles, depending on if we're doing quiet, resting breathing, or if we're doing forced breathing. So if we focus on a relaxed inhale, like the breathing that you're doing right now, There are only two different types of muscles that help us with that, the external intercostals and the diaphragm. If we do a relaxed exhale, it's still those same muscles that are working, but a relaxed exhale is a passive process, whereas a relaxed inhale is an active process. When we use those terms in A&P, remember, active always means that we're using energy, right? So active always uses ATP, which means that during the inhale, we see these muscles contracting. During the exhale, that's passive because they are just simply relaxing. But we clearly know that when, like if we go for a run or something, our breathing gets harder, right? We can do forced breathing. We can force more air into the lungs and out of the lungs than we're doing right now. So we have to have other muscles to help us do that, and that's where these green ones come into play. So during a forced exhale, we're going to see the internal intercostals, as well as all of those abdominal muscles that you studied in A&P1, the external and internal obliques, rectus abdominis, and transversus abdominis. All of those guys are going to contract to help us with a forced exhale. And we'll talk about exactly what all of this contraction and relaxation is doing in just a minute. First, we've got to figure out what our pressures are that we're going to be focusing on, because I told you is going to move from high pressure to low pressure just like the blood did. So let's define a few terms here. Yeah, Randall? Well, think about it. Do a forced inhale right now. You feel your abdominal muscles pulling outward, right? So yeah, so you can feel them working to do that forced inhale as well. Yep.
So, those feelings on the stomach and my stomach during forced inhalation, or what?
They can assist with some of that, I believe.
The stomach
So.
and mastoid and feelings are also muscles of inhalation. So I was wondering, do those come into play during forced inhalation?
Maybe a little bit, but most of it is coming from the abdominals. I mean, if you do a forced inhale right now, you should feel your abdomen changing as well as the ribs changing. Yeah.
To follow up on that then, so for forced inhalation, they also include the external and transverse abdominis.
Yes, and we're going to focus more on the forced exhale than the forced inhale. That's why I have that on the screen. Okay, because I'm trying not to muddy things up too much. So let's go on and let's focus on the pressures. If we're talking about the pressure in the air, in the external environment that's surrounding our bodies, we're talking about atmospheric pressure. And it's important for you to know that pressure, but it's also important for you to know its abbreviation. We'll be calling that PATM. We also have a pressure that we find inside the lungs. What was our fancy term for lungs? Pulmonary. So that pressure is going to be intrapulmonary. Because, well, pulmonary means lungs, and we know intra means what? Inside, exactly. So that name tells you where you find that pressure. Notice they're also showing us that there's a pressure that's coming from the space outside of the lungs. What was that space? The pleural cavity. That's exactly right. So this is the intrapleural pressure. And we're going to have different abbreviations for both of these. Intrapulmonary is P-pul. Intrapleural is PIP. We also have one other pressure that helps us to relate some of our pressures to each other. That's what they're trying to show you with this other line, where it looks like we're comparing pressures across the lung wall. Trans means across. So that's the transpulmonary pressure, since it's happening across the lung wall. And to find that, we literally take the intrapulmonary pressure and we subtract off the intrapleural pressure.
Does this one have an abbreviation?
Most books don't give it an abbreviation because it's more of like a side note that helps us to understand forces that help to keep the lungs open so they don't collapse on themselves. Atmospheric pressure can change depending on where you are. So while we're if you're at sea level, which is where you can kind of think of atmospheric as starting out, it's going to be somewhere around 760 millimeters mercury. If we go up in elevation, that's going to change. Do you know how that would change? It actually decreases. You know how they say the air gets thinner as you go up a mountain? It's because there's less pressure up there. There's less pressure. So you can kind of think of 760 as like your starting point at sea level. That changes if you go up a mountain. And we'll find different pressures for intrapleural and intrapulmonary depending on where we are in the respiratory cycle, whether we're doing an inhale or an exhale. So what the books always have us do is they have us set atmospheric as 0. That way it's easier for us to compare the other pressures to it. So if we were to set atmospheric at 0, if a pressure is higher than atmospheric, it will be positive. If it's lower than atmospheric, it will be negative. So if you see those smaller values that are positive or negative, you're seeing those because they're comparing the pressure to atmospheric. And let's go ahead and make this note right now. Intrapleural pressure should always be negative. Not only is it negative, but it should always be the lowest of our three main pressures here. Intrapulmonary is going to change, though. Sometimes it's positive, sometimes it's negative, sometimes it's even 0. When would we want intrapulmonary to be positive? During the inhale or the exhale? Remember, what does positive mean? All right, positive means that that intrapulmonary is higher than atmospheric, right? So how would the air move that way then? It would move out, right? Because intrapulmonary is inside the lungs. So if that's higher than atmospheric, the air is going to move out of the lungs to the external environment then, right? So we'll see that positive during the exhale. When should it be negative? All right, so it should be negative during the inhale, because that way that's showing us that intrapulmonary is lower than atmospheric, right? So the air can go from the external environment down into the lungs then. The air stops moving when it reaches an equilibrium. When we get our two pressures, atmospheric and intrapulmonary, to equalize. That's going to stop the air movement. So that's when we see intrapulmonary at 0. It's kind of between the exhale and the inhale. Just when we start to switch things up. You guys with me? So intrapulmonary becomes 0. Well, because remember, at 0 would mean that it's equal to atmospheric, right? So intrapulmonary is 0 right between the exhale and the inhale. We're going to see this on a graph too, which might help it to make a little more sense. Sorry, Kaylee, did you have another question?
Oh no, I was like the word you used to say when you reach the...
Yeah, equilibrium. Remember, most things in the body are trying to reach an equilibrium, just like those ions that were moving before, like the sodium when it moved, right? But most things in the body aren't going to actually reach that equilibrium. It's just with this air movement that we see equilibrium finally reached. Okay, and that just means that it finds its balance.
Thank you.
You're welcome. Transpulmonary will always be positive, so let's make a note about that as well. If transpulmonary becomes 0 or negative, that means that the lungs have collapsed. What? For the transpulmonary, that's right. If it is 0 or negative, that means that the lungs have collapsed. Because remember, intrapleural should always be the lowest pressure. And was there another question from you guys back there?
Yeah, I'm still confused why it's positive on why intrapulmonary is positive on exhale.
Well, we'll look at that on the graph, and hopefully that will make more sense, okay? Right. We're going to talk about that more on the graph, though. Don't worry. We'll get there. One other thing while we're looking at our different pressures, we need to make a note about the things that are helping to cause the intrapleural pressure. Because there are kind of some competing forces there that are either pulling the lungs inward or outward to help create this intrapleural pressure. So our one outward pressure is created by the fact that the thoracic cavity itself, that outer wall, has elasticity. That way we can kind of stretch it and pull it open. But there are two inward kinds of pressures. And those are created by the fact that the lungs are elastic like a rubber band, which means that we can pull on them, we can stretch them, but as soon as we let that go, they snap back, they recoil, right? So that recoil is an inward kind of pressure. And also, we're just going to make a note about this for now. I will explain it more later. But we see some surface tension in the alveoli. We'll talk about what that is and what creates it in a little bit. But just to make a note that there's also surface tension in the alveoli, which has kind of an inward kind of pull. So those two competing pulls help to create the pressure that we find in the in the pleural cavity. So that's what creates the intrapleural pressure. What are you confused about? Like I said before, surface, I don't have room on this slide to talk about it. I have another whole slide devoted to that. So just hang out for a second and we will talk about that, okay? Are we good on the pressures? We good on the pressures? Because we're about to talk about how they change. So let's look. I gave you that slide with a lot of the words that I wrote on there, just in case you're having trouble seeing my scribbles. Let's just notice something real quick before we move on. Notice on this labeled picture, they give us both the actual values as well as the values compared to atmospheric at 0. So when we're seeing intrapleural as a negative pressure there, that's the one where it's being compared to atmospheric. Its actual value is the 756 millimeters mercury. Also notice, just like I said, the transpulmonary pressure is a positive value, just like I told you it would be. And they have intrapulmonary set at 0, but we know that can change. Yep.
So how come if that one's the intrapleural is 756, it's only -4?
Well, because if you do the math, we're comparing that to atmospheric, right?
Yeah, but
So if we did the math and we took the 760
never mind.
minus 756, that value is 4 below it, right? So that's okay. All right, let's check out exactly how this is going to work. We'll get to the graph in a second. So we're looking at the inhale first. And I told you that with a quiet inhale, a relaxed inhale, will we see muscle contraction or relaxation? We'll see contraction. Contraction of which muscles? External intercostals and diaphragm, like it shows us on the screen. The external intercostals, when they contract, they pull the rib cage up and out. The diaphragm, when it contracts, it pulls down. If we're pulling the ribs up and out and we're pulling the diaphragm down, what's that going to do to the size of the thoracic cavity? That's going to increase the volume there, exactly. And if we increase that volume, well, that's going to help to pull the lungs open as well, right? Because we know the lungs are going to stretch to follow that movement of the thoracic wall. So that increases lung volume as well. And if we increase the lung volume, what's going to happen to pressure? It's going to decrease which pressure? Intrapulmonary pressure then is going to decrease. And as long as that pressure is below atmospheric, we're going to see the air entering the lungs then, right? With the exhale, it's just the opposite. If it's a nice, quiet, relaxed exhale, remember, all that happens is the external intercostals relax, so they pull down and in on the ribs. And then the diaphragm relaxes, it pulls back up. Well, what's that going to do to the size of the thoracic cavity? It's going to decrease the volume of the thoracic cavity. And if we decrease that volume, what are the lungs going to do? Yeah, they're going to recoil and their size is going to decrease, so their volume decreases as well. And if their volume decreases, what happens to pressure? We see an increase in which pressure? In the intrapulmonary pressure. Good. So as long as we get that up above atmospheric, then how's the air going to move? Out of the body. That's exactly right. Because it always goes from high to low, just like the blood did. If we were to do a forced exhale, well, then we'd need to get the thoracic cavity in the lungs even smaller. So we could increase the pressure that much more to get even more air out. So what muscles were going to help us do that again? Abdominals and the internal intercostals. And will they relax or contract? They will contract. And this is stuff you guys can feel. It's kind of like a cheat. Everybody do this with me. Just do a nice quiet inhale. You should feel your ribs pulling up and out with the exhale. You can feel them sinking back in and down. If we do a quiet inhale, feel right here, followed by a forced exhale, you can feel your abdominal muscles contracting to help with that forced exhale. Okay, so let's put everything together here with our graph. First off, they're showing us pressure and they're showing us volume. Volume's going to be easy, right? Because we know during an inhale or during inspiration, the volume of air in the lungs should go up. During the exhale, the air should leave the lungs, so the volume inside the lungs should go down, right? That's pretty straightforward. But if we look at the pressures, what pressures are they showing us? All right, we can see the intrapulmonary, which where was that again? Inside the lungs, exactly. What else can we see? The intrapleural pressure, which where was that? Outside the lungs, in the pleural cavity, right? And then they're showing us the transpulmonary pressure. They're showing us the difference between the two. So what's missing? Atmospheric. Where should they have put a label for atmospheric? Right at that dotted line at 0. You're exactly right. So if you have your figure with you, you probably want to add that in, that our dotted line right here is atmospheric pressure. Because that's going to help us to understand how things change. So during the inhale, which pressure is the highest? During the inhale? Yeah, atmospheric is the highest during the inhale, right? Because intrapulmonary and intrapleural are both negative. They're both below that 0 for our atmospheric pressure. The intrapleural pressure is being created by those muscles changing the size of the thoracic cavity. Because if we change the size of the thoracic cavity, that changes the size of the pleural cavity. So why is that pressure going down then? What muscles are doing what? The external intercostals, and who else? And the diaphragm, are they contracting or relaxing? During the inhale, they contract, right? Because the external intercostals pull up and out, diaphragm pulls down. How does that change volume? That increases the volume, which is going to drop our pressures.
This is for intrapulmonary.
What?
The
Well, for intrapulmonary and intrapleural, right? Because the intrapleural is decreasing because we've changed the size of the thoracic cavity, which changes the size of the pleural cavity. And then we see the lungs are being stretched. They're being pulled open as well, right? Because that intrapulmonary pressure goes down. The tricky part is that, well, notice those two lines are not exactly in parallel. Intrapleural pressure is going down the whole time, but not intrapulmonary. Intrapulmonary only drops through about half of the inhale, and then it starts to go back up. Anybody know why?
It reaches equilibrium.
But why? Why is it going back up? So, let me ask it a different way. Is changing the size of our container, is changing volume, the only way that we can change pressure? No? How else can we change pressure? Changing the number of molecules inside the container. Think about it. During the inhale, and we're seeing this down here, right? During the inhale, we're adding air inside the lungs. That adds molecules inside of our container that we can only stretch so far. So as we add more and more molecules to the lungs, that's going to bring the pressure back up until it reaches zero. And then we stop seeing net movement of those air molecules. Notice if we look at the exhale, which pressure is the highest now? Intrapulmonary is the highest, so we could make a note of that is higher than PATM there. And we could have done something similar over here for the inhale, except we would have noted that was lower than atmospheric, right? So why is it that the intrapleural and intrapulmonary pressures are increasing during the exhale? All right, we decreased the volume of the thoracic cavity, which decreased the volume of the pleural cavity, and then allowed the lungs to recoil to decrease their own size. What muscles helped us to do that if that was a nice relaxed exhale? All right, I'm hearing diaphragm and external intercostals. Are they going to contract or relax? Relax. They're going to relax. That way the external intercostals can sink back down and in. And the diaphragm, remember, pulls up as it relaxes. So that's what's actually decreasing the volume of the thoracic cavity and allowing the lung volume to decrease in order to bring that pressure up. But notice, we're seeing a similar thing happening where the intrapleural pressure goes up the entire time, but the intrapulmonary pressure only goes up through about half of the exhale. Why is it dropping back down? All right, because we know the number of molecules in the container change the pressure. We literally see less molecules in our container, the lungs. So the fewer we have, the less pressure we will have until we drop that pressure back down to zero where it's in its equilibrium, where it's equalized with atmospheric pressure. Do you guys have any questions about it? So we're feeling good about the graph?
What about the transpulmonary pressure?
Well, transpulmonary pressure, if we were to take the intrapulmonary pressure and subtract off the intrapleural pressure, right, that's going to be a positive difference. The intrapulmonary is higher than intrapleural.
That doesn't matter for the constant, right? It's always negative.
Which one?
The intrapleural.
The intrapleural is always negative, just like it's showing us on the graph, right? It's changing a lot, but it's only changing in the negative range for the intrapleural pressure. All right, well, that's the question that I just asked you to help emphasize that the molecules moving also changes the pressure. And if you're having trouble picturing that, click on the link at the bottom. I don't really have time to do it today, but that's a video that shows you how if we change the number of air molecules in a container, that will change the pressure in the container. All of the purple slides that I gave you are the words that I talked through when we talked through the pressures and the muscle contraction and relaxation that would allow pulmonary ventilation to occur. So that's why I'm clicking through all of those. Those are notes in case you missed something that I said. We do still need to talk about factors that are going to alter pulmonary ventilation. So I want you to make a note about resistance, surface tension, and compliance. While we're sitting here resting, those things really shouldn't change much. But we need to understand them because those are things that can go wrong, especially if somebody gets sick. And also when we talk about how things change when we exercise, resistance is going to change a little bit when we exercise. So since we're seeing this term resistance again, you guys already know the relationship between flow and resistance. What was that equation?
N equals delta P over resistance.
All right, it equals the delta P, or the pressure gradient, over resistance. So if resistance goes up, what happens to flow?
Goes down.
Flow goes down, exactly, because they have an inverse relationship, right? Okay, so you already know that stuff. What you don't know is stuff that's specific to the respiratory system. So make a note that our bigger tubes, like all of those upper respiratory structures, and the trachea, and the bronchi, those are big tubes. They have big diameters. And that, we know, always creates low resistance. Because what is resistance when we're talking about something moving through a tube? What creates the resistance? Friction, right? As the molecules rub against the sides of the tubing. We talked about how moving along the sides of the tubing slowed down the molecules. So the more space you have in the middle, the more room you have for the molecules to move through without rubbing up against the sides and slowing down, right? If you don't remember that, look back at that in your book or look back at it on one of the previous videos because we've talked about that. That point isn't really being demonstrated on this graph, okay? That's why they don't give you any numerical values for resistance. What they're trying to show you on this graph is where resistance is the lowest in the respiratory system. Notice that it's the lowest when we hit the respiratory zone. What was the purpose of that zone? Exchange, exactly. So then why is the resistance so low when we hit the respiratory bronchioles and the alveolar sacs and whatnot? Exactly, because there's no friction. If the molecules interact with the sides of the tubing, they just slip through it. They diffuse through it. So if there's practically no resistance there because the molecules will leave instead of creating friction. So I tell you about all that stuff on this slide. What this slide doesn't tell you is something that I want you to keep in mind for next time. We do see airway resistance changing a bit when we exercise versus when we're sitting here relaxed. So remember how that was regulated with the blood vessels. What did the blood vessels do to help control resistance? They constricted and they dilated, right? Here, since we're not talking about blood vessels, we don't call it vasoconstriction. Instead, we call it bronchoconstriction and dilation. So bronchoconstriction and bronchodilation. And that's why last time I was emphasizing how the tubing changed. Remember, as we moved through the tubing, we saw less cartilage and more smooth muscle. That way the tubing can dilate when we need to get more airflow moving into the lungs. But we'll talk about that more next time. Now, with the surface tension stuff, this is always something that is hard for people to picture. So let me draw a picture to help us understand this. We've cut through one of the alveoli. So we're looking into an alveolus now. We can see its lumen. We know that lumen is mostly filled with what? The lumen, the open space in the middle, is mostly filled with what? Yeah, gas is air, right? That's what we were just talking about with this pulmonary ventilation stuff. However, last time we talked about the fact that that air was modified as we brought it down into the lungs. We warmed it, we filtered it. What else did we do to it? Ooh, this is going to be a long lecture. All right, so remember, guys, last time we talked about the fact that we changed the air, right? The upper tubes aren't just passive tubes that are only letting the air go through it. They filter it, they heat it. What else do they do to it? They moisten it. Thank you. So we add water to the air. And part of the reason that we add the water to the air is because, remember, we have living cells here making up the wall of the alveolus. If you let a human cell dry out, what does it do? It dies, exactly. So it's not just air here. You have to kind of think about it as being, there being like a very thin layer of water that happens right at the edge. And then there's all this air in the middle. And well, water molecules are attracted to each other because if you remember what binds them together, like this one water molecule that I just drew, do you guys remember what kind of bonds those were? They are covalent bonds. What kind? Polar or nonpolar? They're polar, exactly, because the oxygen is more electronegative. So it tries to pull the electrons away from the hydrogens. That makes it a little negative. The hydrogen's a little positive. And we know opposites attract, right? So if we put this water molecule next to another, their opposing charges are attracted to each other. That's what creates surface tension here in the alveolus. And it's not just surface tension with the water molecules right here at the sides next to each other. The alveolus is such a small space that the water molecules across the width of the alveolus are attracted to each other. So it's kind of like they're clinging onto the sides of the walls, but they're also attracted to each other, so it has this effect of trying to like pull the walls inward. That's what they're talking about with the surface tension. And that's a problem because that could promote lung collapse. Plus, the smaller that is, the less air we can get in there to allow for exchange. So we have to have something to fix this problem. And that's where surfactant comes in. We mentioned surfactant last time when we talked about the different cells that we find in the alveolus. Remember, most of the cells are simple squamous epithelial cells, or the type 1 cells, but it's the type 2 cells that make surfactant. And on my slide, I tell you that surfactant's a detergent. So when I think of it, I always think of it as being kind of like liquid dish soap that you would put in your sink to wash dishes. What happens to the water as soon as you add that dish soap? It makes bubbles. It makes bubbles. Well, if we have bubbles, that breaks up the water molecules from interacting with each other, that breaks up our surface tension. So when we think of the alveolus, you can't really think of it as the water molecules just stuck to the sides, because surfactant is kind of creating a bunch of bubbles in here. To reduce the surface tension.
So the water is forming the bubble?
The surfactant, the detergent, right, like dish soap, is interacting with the water to help break it up so it's not pulling those sides inward.
Wait, it breaks up the bonds? Or,
Well, I mean, if we have a bunch of bubbles in there, are we going to be able to have that effect that I was just describing where a water molecule can be attracted to one all the way over here? No, because it's going to be attracted to the other ones that are closer to it in the bubbles, right?
yeah, I want to make sure it helps the ones expand.
Exactly. That's going to make it easier for us to pull the lungs open. It's also going to help to ensure that the lungs don't collapse on themselves. Yeah?
So it's only breaking the bonds between individual water molecules, not
Right. It's not breaking those polar covalent bonds. It's breaking up that attraction between the different water molecules a little bit so that they're not being attracted across the whole alveolus. Yep.
So without surfactant and with the surface tension of the water exerting itself on the alveoli, would that result in lower pressure in the lungs? Leaves are being compressed, or?
That's going to impact the airflow.
Okay. But it doesn't actually affect the pressure.
That would impact pressure as well. But why don't you and I talk about that one-on-one? Because that's like a higher level point than what I'm trying to get across right now. So let's talk about that during the break, okay?
Sure.
Don't let me forget.
Okay.
how do you make it? How is it produced? Surfactant?
That's another thing that you and I can talk about one-on-one. Well, because that's biochemistry. You want to have a biochemistry class right now? Probably not, right, guys? So let's move on, and that's something you and I, because we're wasting time. I don't want to eat up your whole lab time right now. We still have a lot to get through. So for lung compliance, that's the stretchiness of the lungs. That's another one of those things that really should not change too much. We already talked about how as we open up the thoracic cavity, that helps the lungs to stretch and pull open, right? And the more that we open up the lungs, the more that we increase their volume, the more that's going to drop the pressure. We know that the lungs are elastic because we talked about the elastin fibers that we find in them last time. And we also just talked about the fact that surface tension, by reducing surface tension, we help the lungs to be able to pull open more. So those are things that help with compliance or the stretchiness of the lungs. On a day-to-day basis, that really shouldn't change much, but what you have to keep in mind is that the lungs have a delicate tissue. And so if these more delicate tissues in the body become damaged, they don't fill back in with the same type of tissue. They fill back in with scar tissue. And that scar tissue is not going to be as elastic. So if someone has a pulmonary disease where their tissues are becoming damaged, that's going to reduce the compliance of the lungs. They won't be as stretchy. The more scar tissue they have, the less stretchy they will be. Also, if those type 2 cells stop making surfactant, that will increase our surface tension, which will decrease the compliance. One last thing about this compliance business. We know that changing the size of the thoracic cavity changes the volume of the lungs, right? So if something happens like paralysis, where the muscles are not able to act to change the size of the thoracic cavity, then we can't pull open the lungs as much either, right? Because that is dependent upon changing the thoracic cavity. Eden, did you have a question?
No, I did, with regards to the scar tissue on the lungs. So that never gets healed.
No, it's a similar effect that happens during heart disease as well. That's delicate tissue. When it's damaged, it fills in with non-elastic scar tissue, which decreases its stretchiness. Mm-hmm. Yeah, Randall?
Can decrease flexibility of the thoracic cage because shallow breathing,
What do you mean?
If you don't take deep enough breaths, it just tightens up, so you're not able to expand as much if you don't do any deep breathing.
It can happen with muscles. I see where you're coming from there. Huh?
Not kind of
That still would happen in more of a diseased kind of state, right? Because it's not like we're consciously thinking about how much we have to contract those muscles in order to do the inhale and the exhale, right? Before we take our 10-minute break, I'm going to let you have one in the middle of this lecture because it's going to be a longer one. But before we do that, we're not quite at a really good stopping point yet. So I want us to talk about lung volumes first and how we measure them. And then we'll take our break, and then we'll come back and we'll do exchange. Thank you. So a few things before we look at the actual lung volumes. You do need to know respiratory rate. You need to know that we measure it as breaths per minute. And while we're sitting here resting, that should be somewhere around 15 breaths per minute, give or take 3. You also do need to know tidal volume. This is one of the things I'm going to show you on a graph in a minute. You do need to know that that is 500 milliliters per breath for the typical adult. And when we say breath, that means during a nice quiet inhale or a quiet exhale, we will see 500 milliliters of air moved. We have a term that relates these two terms together. That's what minute volume is. So that's the volume of air that the lungs move every minute. And that is one of the better indicators for figuring out lung health. If we look at lung volumes, that can be a first indicator to let us know if maybe there's something going wrong. So let's look at those volumes.
So is there like an equation for the minute volume?
There is, but I'm not making you memorize it, so that's why I didn't have that on the slides.
Okay.
Yep.
Just to clarify, inhale or exhale.
Mhm. Yeah, and I'm going to talk about that on this graph too. So looking at this graph, let's just look at what the graph shows us first. Let's not look at the terms for the volumes yet, okay? We're seeing volume in milliliters, and we're seeing that measured over time. I know they don't have that labeled, but that's what we're seeing on the x-axis. So let's see what this person did. They did a nice quiet inhale, followed by a quiet exhale. Then they did a forced inhale, followed by a forced exhale. Then they did another inhale, followed by more quiet breathing. Okay, so that's what it's showing us. Tidal volume of 500 milliliters, notice, shows us the volume of air that's moved during one full inhale or one full exhale during that quiet, relaxed breathing. Right? Because it's changing between about 2,500 and 3,000 milliliters. That's a difference of 500. Inspiratory reserve volume is the volume of air that can be forcibly brought into the lungs past a normal inhale. So the inspiratory reserve volume is the volume of air that can be forced into the lungs past a normal, quiet, relaxed inhale. Expiratory reserve is just the opposite. That's the amount of air that we can move out of the lungs during a forced exhale beyond what we would normally move out of the lungs. What's going on with that residual volume? What is that? Yeah, that's the air that's always going to be there. That's the air that we cannot remove from the lungs no matter how hard we try. So just like we talked about with the ventricles and the heart, remember when they contracted, they got a lot of the blood out, but they didn't get all of the blood out? It's a similar deal with the lungs. We're going to keep some of the air in there. That way the lungs don't collapse. Okay? On this graph, they're also showing us capacities. So you do need to know that a capacity relates at least two of our volumes. So the inspiratory capacity is literally the volume of air that we can bring into the lungs after a nice, quiet exhale. So notice that is involving both the inspiratory reserve and the tidal volumes. Functional residual capacity is the volume of air that's typically left over after a nice quiet exhale. So that's the residual volume plus the expiratory reserve. Vital capacity is the total volume of air that can be moved. Like if we were to do that forced inhale followed by a forced exhale. All right, so that's what they're showing us right here. Total and capacity kind of speaks for itself. That's all of the air that we can find in the lungs, regardless of whether it moves or not. Yeah?
For the one that stays in the lungs, it's always cleansed. It's the same volume, but it's different, right? It's not like it's the same air.
We're going to talk about that in a minute. There's kind of a mixing effect that happens when we do that next inhale, where we mix up the new air with that old air that's left over. That's a good point. That's going to be really relevant after our break. Yep.
The air that stays in your body doesn't has to be lower.
What?
I just missed that. The air that stays in your body, like when you exhale.
No matter what? Even when you try to force the most out? Well, you guys help me out with that. Which volume is always going to be there no matter what? Residual volume, because even after a forced exhale, where we've gotten all the air out that we can, we've got that residual volume left over. A couple of other things before your break. The book gives us a nice table to go along with the graph. You do not need to memorize every single value in that chart. You should know the average values that they talk about in the graph, but notice those values really just relate to who?
The person first. No, male.
Males. How do most of the females change in comparison?
Less.
Female values tend to be lower. Why?
Because they're smaller.
Exactly. Smaller bodies have smaller lungs. The typical female is going to be smaller than the typical male. That's why they're showing you different values for the averages for the female. Except for which one? Which one's the same?
Sorry.
The tidal volume, right? Tidal volume is still 500 milliliters, typically, for males and females. Dead space is something that we also need to note while we're here thinking about the air in this pulmonary ventilation. Not all of the air is going to make it down into the alveoli. When you do a maximal inhale and you get as much air into the lungs as you can, there's still going to be air backed up in areas like the trachea and those bigger bronchi where it's not going to be used for exchange. We call this air dead space because it's brought into the body, but the body doesn't really use it. Also, since we were saying these volumes can be used to indicate if maybe there's a problem going on with the lungs, I want you to know about one of the tools that we can use to measure lung volumes, the spirometer. And that's what I'm showing you in the picture here. Notice there's a little tube that you can blow air into, and when the air is blown into it, the volume of air is measured with that gauge. Okay, kind of like with the blood pressure cuff, except that this will be measuring in milliliters, since we would be measuring volumes there. Remember, I told you that measuring volumes might give you some evidence that there's a problem with the lungs, but that's not the be-all, end-all. That just means that more tests need to be done. There are lots of different tests that could be done, but since we mentioned the minute volume, you might want to make a note that that is a specific example of something that could be measured to help assess lung health. This is where I'm going to pause us. Let's take just a 10-minute break and come back in and finish up this gas exchange stuff, and then we'll talk about labs. So go ahead, get up, move around. So we've still got to talk about internal and external respiration. That's what we're focusing on when we focus on gas exchange. So the setup here is going to be a little bit different. With pulmonary ventilation, we were talking about all of the air molecules moving in the same direction, going into and out of the lungs, right? But with respiration, like with the external respiration, let's focus there first. So who's exchanging there? What's the exchange is always with the blood, right? So with external respiration, who is exchanging with the blood?
Lungs.
The lungs, the alveoli, right? In the lungs. And if we focus on our two major gases that we care about, oxygen and carbon dioxide, would we want both of those gases moving in the same direction here?
No.
No, we wouldn't. Who's going to be going into the blood here? Yeah, oxygen will be leaving the alveoli and going into the blood, whereas carbon dioxide is going to do just the opposite, right? So we kind of have a problem. We can't just focus on overall pressures because we have two different molecules that we need to have moving in two different ways. And in order to understand that, how that works, we have to understand partial pressures. That's what Dalton's law tells us about. So, the air surrounding us is made up of different types of molecules. It's mostly nitrogen. And if we think about the fact that, pressure is created by molecules whenever they interact with things, right? If nitrogen makes up close to 70%, so close to 80% of the molecules in the air, it should be making up about 80% of the total air pressure, right? Okay, it's a similar deal if we look at the other molecules. We have a fair amount of oxygen, close to 20% of the molecules in the air in this room right now are oxygen molecules, so they're going to contribute 20% to the total pressure. We have a bit of water vapor, and we also have a bit of carbon dioxide. And each type of molecule is creating a pressure equivalent to its percentage. If we were to add up all of the pressures here, that would give us our total overall pressure. Now, these smaller pressures that are being created by the individual molecules, like that 159 for oxygen, that's called its partial pressure. And when we talk about partial pressures, we're going to have abbreviations. So if I wanted to talk about the partial pressure for oxygen here, I would call that the PO2. If I talk about the partial pressure for carbon dioxide, that's going to be PCO2, and so on and so forth. So, the nice thing is that the setup here is going to be fairly similar to what we've seen before. We've always seen stuff moving from what to what? From high pressure to low pressure, except now we're focusing on specifically the partial pressures. So like we'll see oxygen moving from the area where it has a higher partial pressure to the area where it has a lower partial pressure. And that is what Henry's law tells us about. Okay, that these molecules are going to be moving along their partial pressure gradients, especially if we're looking at an interface like between air, gases, and liquids. So like if you think about where the air in the alveoli is going to be exchanging with the blood in the capillaries of the pulmonary circuit. So just to help us kind of visualize how this works, let's take our little person and all of those color-coded air molecules, and let's just focus on oxygen. So let's get those other molecules out of the way. Now notice I've put him so that he's walking on water, so now we have air coming into contact with fluid. And where do we see a higher partial pressure for oxygen, in the air or in the water? In the air, right? Because there's more oxygen in the air. So then, how would we expect those oxygen molecules to move for their net movement? Will we see a net gain in the fluid or gain in the air? Gain in the fluid, exactly. The tricky thing is that this partial pressure gradient is not the only thing that regulates movement of air molecules. Solubility is also a factor. And I'm not going into the chemistry of all of this. That's one of those things we're saving for your upper level classes. But I need you to keep this in the back of your mind, because carbon dioxide is way more soluble than oxygen in water. And because it's so much more soluble, it doesn't need a pressure gradient that's as steep as what we're going to see for oxygen. So when we look at the actual pressure gradients in just a minute, we're going to see a bigger difference in the partial pressures for oxygen than we will for the partial pressures of carbon dioxide. But we see just as many carbon dioxide molecules moving because its solubility is different. Before we look at how this respiration is actually going to work, we've got to take a moment to think about how we have changed the air. So notice in the table that I'm giving you, we have pretty much those same values in the atmospheric air, but now I'm having you compare those to values that we find in alveolar air. So in the alveoli. What goes up? All right, water vapor is definitely higher in the alveoli than it was in the atmosphere. What else? Carbon dioxide is also dramatically different compared to what we saw in the atmosphere. What goes down in a significant way? Oxygen levels definitely go down, right? Why do we see these differences? What's creating these changes? All right, so for one thing, we know that exchange happens here in the alveoli, right? Where oxygen is leaving the alveolar air and going to the blood. So that kind of helps to explain why those levels are lower, right? And why carbon dioxide levels are higher in the alveoli, because that's being added by the blood to the air there. So exchange is one factor. What's another factor? Like for the water vapor. How did we change the air, guys? We moistened it. So we added that water to the air as we brought it through all of that upper tubing down into the lungs. There's one other thing that helps us to understand why this is different. Did we remove all of the air from the lungs with that maximal exhale? No. What do we call that volume? Residual volume. Because of the residual volume, that leftover air after every exhale, right? Think about it. Every time we inhale then, we're bringing in fresh air that's going to get mixed with the old air. And so that's going to amplify all of those other effects that we just talked about, because you never totally get rid of that extra carbon dioxide or water. You guys with me? Or have questions? Yeah, I gave you the words for it. So, we add water to the air, we moisten it, and we have exchange happening, right? So that explains on a kind of a basic level what's happening with the increase in the water and increase in carbon dioxide, decrease in oxygen. But also you can't forget about the fact that we don't remove all of this air that we've used from the lungs. When we exhale, some's left over. So that old air, the used air, gets mixed up with new fresh air every time we do an inhale. Notice if we look at partial pressures for oxygen and carbon dioxide, We're seeing similar trends for atmospheric air and alveolar air here. PO2 goes down in the alveolar air, right? CO2 goes up compared to atmospheric. Also notice they've basically set up this figure in two different loops. The first part, right up here, is going to show us what happens in the pulmonary circuit. Or in other words, where we're seeing that external respiration, right? And then down here, we're going to see what happens in the systemic circuit, or with the internal respiration. And in order to think about how the molecules are moving, we've got to focus on the blood that's coming to the area as well as the area itself. So if we want to see how external respiration works, we've got to compare the partial pressure values in the alveoli to those pressures in the deoxygenated blood that's being brought to the lungs. If we do that comparison, where do we see a higher partial pressure for oxygen? In the alveoli, exactly. So how's oxygen going to move? Yeah, it's going to move into the blood, right? That's what they're trying to show you right here. So the oxygen is leaving the alveolus and getting into the blood because it has a lower partial pressure in the blood. It's going from high to low. What about carbon dioxide? Where's its partial pressure higher? In the deoxygenated blood that's being brought to the lungs. So that means carbon dioxide is going to leave the blood and get into the lungs then, right? Notice if you compare the partial pressure values, the difference for carbon dioxide is almost nothing, especially compared to the difference for oxygen. But yet I told you that we see the same number of carbon dioxide molecules moving as we do oxygen. Why was that again? Because of the solubility. Who's more soluble? Carbon dioxide. So it does not need as steep of a gradient. If we were to do a similar kind of comparison and look at the systemic circuit, we need to compare the partial pressure values that we find in the tissues to the blood that's being brought to them, so the oxygenated blood. And in this case, where are we going to see a higher partial pressure for oxygen? In the blood, exactly, right? 100 is definitely bigger than 40. So how's the oxygen going to move? Yeah, out of the blood and into the tissues. What about carbon dioxide? Where's its partial pressure higher then? Yeah, in the tissues. That way it can leave the tissues and get into the bloodstream where it has a lower partial pressure, right? The other thing that I need you guys to look at on this diagram is how the blood looks when it leaves an area. So if we look at the deoxygenated blood that's leaving the tissues, and we compare those partial pressures to the partial pressures that we find out there in the tissues, notice that they're the same. They reach their equilibrium. Kind of like what we saw with pulmonary ventilation, right? Where everything moved until we reached an equilibrium. It's a similar deal when we look at the partial pressures. Oxygen and carbon dioxide are going to move until they get at least close to their equilibrium. We can see a similar effect if we compare the air in the alveoli to the oxygenated blood. Notice the partial pressures are almost the same. They're just about at their equilibrium there. Any questions about internal or external respiration? All right. Next time we'll be talking about transport. We'll talk about regulation of processes, and we'll see how things change when we exercise.